Hi, welcome to the vodcast on DNA structure. Today's vodcast will talk to you about how DNA is actually built, and by you understanding that, that'll allow you to actually understand things like genetic engineering, stem cell research, and cloning as we start to talk about it. So let's get started. Just a friendly reminder, your notes need to be in Cornell note format. So remember, leave yourself a good 2-inch margin on the left, leave yourself some space in the bottom of your page 2 for a summary statement. So let's get started. So to give you a quick idea just on how much DNA you have inside your body, not just one cell, but every cell in your body has DNA. If you were to stretch that out, you could go back and forth between the Earth and the Moon about 6,000 times. Now granted, you wouldn't be able to see this strand because the strand would be so ridiculously small, we'd be measuring it most likely in things like nanometers, not millimeters, but nanometers. So, continuing through. So title for your presentation here will be genetics. And basically, why do we look the way we do and how are we passing these characteristics along? So let's start out with a quick review of what is genetics. And we've been over this before, but basically remember, genetics is the study of DNA and traits and how they get passed on from each generation. Remember, you're getting your parents' DNA. You get one allele from your dad, one allele from your mom. Now, DNA, we always use the short term, and if you ask most people on the street, they couldn't tell you what the heck DNA stands for. They just know what it is. So it stands for deoxyribonucleic acid. It looks a lot worse than it actually is. You can all say it easily at home. If you want to seem a little weird, try to say it out loud and see what happens to people walking by your room as you're going, deoxyribonucleic acid. All right, so what is it? First off, let's go with the structure. We refer to the general structures you'll see over here as a double helix, all right? And they get that name because a helix literally means a spiral. And since you'll notice there's two of these spirals, all right, we call it a double helix. So we also commonly call it a twisted ladder or a spiral staircase. So we have 6 compounds. You would think the thing that makes us who we are would be way more difficult. When researchers try to figure out what DNA looked like, they thought it was going to be this crazy, crazy thing, and they were really focusing in on the amino acids that DNA can make rather than the DNA itself. DNA itself is really simple. So you've got a couple things. First off, it's a ladder, right? So you've got the outsides of the ladder, and you've got rungs. The outside of the ladder is made of something called sugars and phosphates. Sugars, you're well aware of what they are. That's where the deoxyribose comes from, all right? Deoxyribose is actually a sugar in the body. The phosphates also join the sugars together and make up the sides. Then you've got the bases, or what we would refer to as the rungs of the ladder. These are the actual nucleic acids, all right? And they pair up, just like we talked about in the video. A and T pair up in equal amounts, and C and G pair up in equal amounts. And my computer just keeps making karate sounds for absolutely no reason. There we go. Okay, so who discovers DNA? Rosalind Franklin, first off, is key. She develops this X-ray crystallography picture, all right? She's the first one to really get a good, clean shot. When you look at this piece right here, this X design, if you're looking at a double helix, where the helixes cross over, they're going to form an X. All right, and that's why that produced that picture. She uses X-rays to produce these images. The downside is it also leads to her getting cancer and dying at a very young age. Very uplifting, I realize. Now, Watson and Crick are kind of like two goofballs who know a lot of stuff, a lot more than people give them credit for. So these are the scientists who built the first model of DNA, showing its actual structure and bonding. All right, and here's these two young chaps, and they've got their crazy metal model of DNA, because back in the time period, you built it all out of metal or cardboard. Those were your options. There was no plastic. Now, who likes who? How do you remember what bases go together? I'll give you some hints. Okay, the only way they go together is A and T, C and G. You can't mix A and C. You can't mix A and G. It doesn't work. First off, it's sizes, okay? The sizes of the molecules have to match. Otherwise, your ladder would go really wide and then really narrow, and that wouldn't be a really good stable ladder. The other problem is chemically, they cannot bond any other way. They just physically don't have that ability. So you get A, T, C, G. If you can't remember which one pairs with which one, use my hint at the bottom of the page. At cross gates. A, T, C, G, right? A and T go together, C and G go together. So at cross gates. Otherwise, if you put them in all in alphabetical order, A, C, G, T, the A and the T, the farthest ones away from each other, go together, and the ones in the middle will go together. So whatever hint works for you as far as that's concerned. Okay, what are genes? We've talked about this before. We've talked about this a lot in your fruit fly labs. Genes are sections of DNA that code or cause a trait. So basically, it's like instructions. All right, and most of these instructions make proteins. So you'll see a 3-letter sequence. So every 3 letters, 3 letters, 3 letters, they code for amino acids. And those amino acids make up proteins. I always like to say it this way. Think of these 3-letter sequences as Lego blocks. You stick enough Lego blocks together, you build a house, or you build a boat, or you build a car. In biology, if you stick enough amino acids together, you build a protein. All right? So one of the things we haven't done yet in class, but we'll do relatively soon, is giving you some examples of some traits. Not all trait is as simple as eye color, hair color, skin color, or anything like that. Some of the traits you have are actually inside your body. How you taste your food can actually be a genetic trait, just like tasting PTC. All right. So here's 2 questions I want you to write down your notes, and I want you guys to answer these. All right. And I'm going to check them in your notes. Here's what I want you to do. Question number 1 says, what base pair with this half strand of D? So underneath these letters, on the bottom row here, I want you to write what letter would go with A. So I'll do the first one for you. A always has to pair up with what? It's got to pair up with T. So underneath the A, I would write a T. T, therefore, always goes with A. So underneath this T, I would write an A. So I want you to complete the rest of this strand. Number 2, go back to previous notes you took in this vodcast. What other compounds are found in DNA besides adenine, thymine, guanine, and cytosine? All right, so I want you to go back, and I want you to figure out, there's 2 of them. Give you a hint. There's 2 more compounds, and they make up the sides of the ladder. All right, I'm going to check those when I check your homework. So hopefully you guys got what you needed. If you still don't understand this, see me for help, and we'll help you out. We'll be doing DNA extraction and a couple other labs, some DNA keychains, and we'll go from there. All right, guys, have a good night.